Hello guys and good day. This is our lecture for our Module 5, Nucleic Acids. Okay, ano ba pag-uusapan natin dito guys? Pag-uusapan natin dito guys is the DNA structure, the nucleotides, and the simple problems about the central dogma of molecular biology, which the transcription and translation processes, which we will discuss later. Okay. So ito I just providing a simple lecture just to para lang maging familiar kayo sa concepts of nucleic acids. But the in-depth topic of this will be discussed in your lecture class. Pero supposedly kasi dapat tayo ay puro laboratory lamang sa magaganap sa ating classes. But before that, I'll just provide you the basic information or the concepts about nucleic acids. Okay. Nucleic acids, guys, or the deoxyribonucleic acids and DNA carries the genetic information in the cell. Saan ba nakalocate ang DNA natin? The DNA, the chromosomes is made up of DNA, guys, by which the chromosomes is located in the nucleus of our eukaryotic cells. And yung ano naman natin, sa cytoplasm naman na-allocate yung chromosomes ng ating mga prokaryotic cell. Okay, ang DNA is a molecular archive for protein synthesis na kung saan magsisynthesis siya ng ng RNAs, magta-transcript pala in order yung RNA natin which is responsible to translate those RNAs into proteins. So ang natin, DNA is comprised of two polymer strands na ang intermolecular forces na nag-hold on is the hydrogen bonding. That is kaya meron siyang twisted ladder dahil nga dun sa may hydrogen bonding. Meron niyang, I think, 3 to 5 angstrom per base pairs per twist. Ganon. Tapos, the sides of the ladder are alternating sugar and phosphates unit na kung saan naman yung namang kapitan or the rank of the ladder are the hydrogen bonded pairs of our heterocyclic amine bases. So, ano ba yung mga heterocyclic amine bases natin? Okay. The sequence of heterocyclic amine bases in the DNA encodes the genetic information required for protein synthesis. Okay, only 4 different bases are used for the code in DNA. Apat din naman sa may RNA. By which DNA, ano ba yon? Yung AGTC natin, the adenine, guanine, thymine, and cytosine. While the RNA naman natin, guys, is AGCU, adenine, guanine, cytosine, and uracil. And then the section of DNA that encodes for a specific protein is called a gene. Mamaya mako natin yan. Ayun, kung mag-discuss naman. And the set of all genetic information coded by the DNA in our organism. So, kabuuan na siya, guys. It is, tawag doon, is genome. So, this is a schematic map of the chromosome 19 na may kita sa ating cells natin. And you can see the color, which are the responsible dito sa mga base pairs natin. Composed of 59 million base pairs kasi yung chromosome natin, guys. Na kung saan ito yung map natin, yung mga colors nito is, yun yung mga indicator na magkakaroon tayo ng mga disease natin. Diba nga, kaya nga sinasabi, is related daw. Yung daw mga other diseases are hereditary. Kasi nga ang responsible doon is yung mga chromosomes base pairs na ma-inherit natin sa ating mga magulang, di ba? 23 sa tatay natin and 23 sa nanay natin. So, mga ganun-ganon. So, yung mga natin is mapapasa. So, kaya ganun. Kaya hereditary yung ibang genes natin. Dahil nga, it contains some of the chromosomes. So, mga ganun-ganon lang. I think you are all familiar for that. And then, kapag daw nagkaroon ng maldegradation ang nucleic acids, magkakaroon siya ng yung monomer units na nucleotides. So therefore, nucleic acids is a monomer of nucleotides. And then further hydrolysis of nucleotides comprised of 3 components. Those are the heterocyclic amine bases or the AGTCU. AGTC kasi ayon. And then the other one is the D-ribose. Okay? It is a 5-membered carbohydrate ring in terms of heart projection from RNA. Kapag naman sa DNA is a 2-deoxy-D-ribose na kung saan doon ay makikita doon sa mawalan siya doon sa may carbon number 2, I think. Yata. Mamaya makikita natin. So, and then a phosphate ion. So, ganyan, do nucleic acids. I-discuss natin nucleotides, DNA structure, and the RNA. But we will not go on to the secondary structure, tertiary interactions, and the wobble hypothesis, yung mga diagrams sa mga DNA replications, mga ganun-ganun bagay. Punta tayo sa nucleotides natin. Okay, as we can see here, guys, dito, sa ating phosphate, okay, it composed of, ito yung ating bases, adenine bases natin. Then the sugar, which is the D-ribose. Dito naman, it is a deoxyribose kasi nga, ayun, carbon number 2 pala, guys. So, 2-deoxyribose na nakasulat din naman pala dito kanina. So, carbon number 2, wala siyang OH doon. Sa RNA natin, merong OH. and then phosphate. Okay, punta na natin. Ang nucleotides are found primarily as the monomeric units. So it is the monomeric units of nucleic acids of the cell of RNA and DNA. Ano ba yung nucleotides natin? Ito yung structure niya. Okay? Ang functions niya, guys, is serving as an energy storage, meaning ATP or the adenosine triphosphate, yun nga. And then, nag-control siya through enzymatic reactions through allosteric effects of enzyme activity, which are the enzymes madidiscuss yun sa may part ng protein discussion ng inyong lecture class. Pero that's not our interest for today. We will focus on the mga nucleic acids natin. And then it is also a mediator of numerous important cellular processes such as second messenger in signal transduction events. So series of enzymatic chemical reactions in our body. So it is an mediator, yung mga nucleotides natin. Okay, now our nucleotides is comprised, as discussed earlier, is a purine or pyrimidine bases, a pentose, specifically ribose or deoxyribose kapag sa DNA, and then a phosphate group. Phosphate group. Okay. So, ganyan. Unahin muna natin, guys, yung ating purine bases. Okay. There are two types of bases, guys, sa ating purine or pyrimidine base. Sa may ating heterocyclic amine bases, guys. It is purine and pyrimidine. Kapag yung purine natin, guys, para mas minimize is pure Ag or pure silver. So, adenine natin, ang purines natin, kapag purines, that is adenine and valine. Kapag pyrimidines naman, that is pwede rin cat, pyrimidine cat pyrimidine. Kasi ngayon ay mga CTU. And these are their common structures. Yan yung purines, itong mga to, sa purines natin, magkakaiba dyan kung saan nakalakip yung amino group and yung carbonyl group natin. Doon sila magkakaroon ng variation. Okay? So, ito naman, other one is the ribose. Ano ba yung ribose natin? Ang ribose na natin dito is a 5-membered ring. Ito, 5-membered ring or 5-carbon sugar na na-discuss natin sa carbohydrates natin na na-project into Howard projection. Hindi naman siya dito yung form natin sa anyang nucleotides natin. But rather, siya ay naka-Howard projection. Naka-enclose in 5-membered ring. So, ito yung natin, guys. Yung ating amine natin ay amine. So, yung purines natin are Ag. Ito yung structure ng ating adenine. Okay? Ito yung adenine natin. Kapag kaya adenosine monophosphate yan, guys, kasi nga kapag yung phosphate natin, yung adenine natin is na-bond doon sa may nucleotides natin na may isang phosphate group. Okay? Kapag triphosphate, 3 phosphates yun, guys. Okay. Ito yung structure ng ating adenine. Ito, adenine. Ito, ganito. Okay. Ito siya. Yung carbon, yung amino group is nandito sa may 6 carbon natin. Kasi ang pag na 1, 2, 3, 4, 5, 6, 7, 8, 9. And then dun sa may 9 nitrogen natin, guys, is doon nakaband yung ating, doon siya nakaband sa may natin, sa may ribose natin. Okay? Next naman natin is palitan ninyo dun sa may 6 carbon ng double bonded O. And then dito sa may third carbon sa may second carbon natin is lagyan niyo ng amino group. Ayun ay magiging guanine na. Pero parehas lang din yung naaban sa nila sa X. Sa may pentose sugar natin, kung saan naaban siya sa may ating carbon number 1 ng ating ribose. And then for the CTU or CUT, okay, for the cytosine, ito yung carbon number niya, guys. Magsisimula tayo dito pa clockwise. 1, 2, 3, 4, 5, 6. Ngayon, kapag kinan natin siya dito, sa may fourth carbon, meron tayong amino group, and sa may second carbon, meron tayong carbonyl group for cytosine. Kapag both carbonyl group sa may 2 and 4, okay, that is uracil. Kapag naman meron tayong carbonyl group, and then methyl group sa may fifth carbon, ayan, that is thymine. So, CUT. Okay, amino, carbonyl. Dalawang carbonyl, isang methyl. CUT. Okay. And ito yung mga pangalan nila kapag sila yung nucleoside. At ito na yung pangalan nila kapag sila nasa nucleotide. Sir, what is the difference between nucleotide and nucleoside? Kapag nucleoside, guys, is wala tayo phosphate group. Okay, wala tayong phosphate group. So, ganyan. So, these are the base conformation of our specific nucleotides here and nucleosides to, nucleoside to. Okay, kasi wala siyang phosphate group, nucleoside to. Ang kwan natin guys dito is, ang nagde-dominate na conformation dito is the anti. Ito yung anti, ito yung syn. When we say anti, ito, nasa opposite direction siya with respect to our ribose. Kapag namang syn is, nasa same orientation siya with respect to the adenosine natin. So, nagde-dominate dito guys is the anti-conformation. Kung nare-recall ninyo yung pinag-aralan sa organic chemistry, ito ay mga specific conformation of our organic compounds sa mga natin, sa mga alkene conformations natin. So, anti-predominates. Kasi nga, kung titignan natin is, meron silang stability. So, less hindered or less steric steric hindrance. Wala tayong steric conformation. So, mga ganun-ganun bagay. Okay? Okay, the sugar conformation, ribose sa flexible ring. Ito, okay. Mga conformations lang yan. This one is C3' endo and then C2' endo. When we say endo, yun yung nakatakas. So it's just a conformation. The sugar conformations natin. Okay. Sa may mga polymerase nucleotides naman natin, guys, kapag nilink nyo siya, guys, it is a 5' to 3' chain. Okay? 5' to 3' chain. And then ang nagko-connect sa kanila is yung ating mga phosphate group. Okay? So ito, kung ang nag-link sa ating carbohydrates is a glycosidic bond or glycosidic linkage, sa nucleotides naman natin is phosphodiester linkage ang nag-link natin. Kasi nga, di ba, ang nucleic acids natin comprise of monomeric units. Okay, so ayun, phosphodiester linkage na kung saan nakalocate sa may 5' to 3'. Sir, what is 5' to 3'? Kasi nga, ito yung carbon natin. Carbon number 1, then our base, number 2, number 3, so number 3, number 4, number 5. So, 5' to 3'. And, ito, tingnan nyo. 5' to 3'. The 3' is connected to 5', and the 5' is connected to 3', and so forth. And ito na yung pa natin. So, phosphodiester linkage. And as you can see here, the difference guys in DNA natin, deoxyribose at the second carbon ng OH. And yung RNA natin is, ito, meron siyang hydroxyl group sa second carbon natin. And sa RNA natin is also 5' to 3' with link with a phosphodiester linkage by which the phosphate group links our nucleotides. Okay, pag ganun siya. So, kung pagdodrawingin kayo guys ng mga phosphodiester linkage natin, mga ACCCCGGTTT, mga ganon, is pag ganun siya. Pa 5' to 3' with ito. Ayan. Ang nag-link sa niya is phosphate group. So, mga ganon. 5 carbon to 3 carbon, 5 carbon to 3 carbon. So, mga ganon. And then, the nucleotides natin is an energy carriers, guys. Kasi nga, di ba, is a composed of, consider this adenine. Anong pangalan niya? Meron siyang 3 phosphate group. Siya ay ATP. So, adenosine triphosphate. Kapag dalawa lang, diphosphate or ADP. Kapag isa lang yung phosphate, AMP, adenosine monophosphate. Ito, nandito pala siya. Okay. So, mga ganyan. That is the main energy exchange reactions in cells by which mapapag-aralan ninyo sa nucleotide metabolisms kung ma-discuss yun sa inyo lecture ninyo. So, puntahan natin yung DNA structure. Kasi we're done with the nucleotides. Diba, nucleotides, RNA, DNA, comprised of a heterocyclic amine bases, sugars, which is the ribose, and linkage, and phosphate group, by which the nag-link sa kanila is phosphodiester linkage with the help of our phosphate group. Okay, the DNA natin, guys, is a double-stranded helix as described here nga. Ito, as you can see from the model, the structure is basically a long, twisty, rope, laddery type thing. So, ganyan. This is ko natin. So, it is a double-stranded helix in DNA natin na kinuha natin. Ang nag-propose ng model na ito, na kanila, isang nag-predict nito ng structure is si Watson and Crick, na originally ay si Rosalind Franklin. Pero sila yung na-credits. Okay? Sila yung nag-publish na ganito daw. So, double-stranded daw yung ating DNA structure. Na ito, na kung saan ito ang nag-link sa kanila. Ang nag-link doon, kaya nagkaroon ng double-stranded base, is yung mga hydrogen bonding natin sa adenine and thymine and G and C. Okay, kung papansin niyo na dito, ayun o, sa may kung mapapansin niyo guys, di ba, kapag nagkakaroon tayo ng mga base pairs, ang katapat lagi ng adenine is always a T. Di ba? Kapag sa may opposite side. And then sa may guanine natin ay cytosine lamang. So bakit kaya ganun? Kasi nga, doon nakadepende yun in terms of compatibility in their hydrogen bonding. Yung adenine natin, comprised of 2 hydrogen bonding. So bakit hydrogen bonding? Kasi nga yung H natin is nakikipag-interact sa N ng adenine. And then yung H ng adenine is nakikipag-interact doon sa may carbonyl group or oxygen ng ating thymine. Whereas our guanine and cytosine here is ito, sa may sugar natin, and sa may natin, mga base natin, is yung, ayan no, meron tayong 3 hydrogen bonding there. So ayan, i-quote ko lang, in other words, if an adenine forms one member of what pair on either chain, then on these two assumptions, the other member must be thymine. Similarly, for guanine and cytosine, the sequence of bases on a single chain does not appear to be restricted in any way. However, if only specific pairs of bases can be formed, it follows that if the sequence of bases on other chain is given, then the sequence on the other chain is automatically determined. So mamaya magka-aaround tayo ng mga complementary strand. Okay, so mga ganyan, major group. And then ito, another quotes and excerpts from Watson and Crick. So ganyan. Kaya na-device nila. Ayan. 5' to 3' end. And then yung complementary niya guys is 3' to 5'. Ganun lagi. Okay? Kapag nag-1, 5' to 3', hindi siya pwedeng 5' to 3' end kasi nga complementary yung kinukuha natin dito guys. So dapat ang complementary 5' end natin is a 3' end. Okay? So ayan, 3.4 angstrom per base pair and 10 base pair per turns. 10 to 11 yan aqueous solution and 2 anti-parallel strands. So ayan, sinukat yan from the lineager and fifth edition. So that is 10 base pair per turn. So these are the natin, yung other DNA strands natin sa mga locations natin such as Perth, Australia, Life Science in UC Davis, and I think that's Cambridge. Siguro, yun nandun. Okay. Some of the DNA structures. Ayan, specific pairing. Ngayon, punta na tayo guys sa ating replication of DNA. Hindi natin i-discuss yung mechanism nito guys yung DNA helicase, yung pag-unwind ng chain, the leading strand, the lagging strand, mga ganun. Hindi natin i-discuss yun. Pero, kunin natin yung only the surface of its topic. Okay? Ayan, so ito, the DNA strand begin to unwind just prior to cell division. Complementary strands are formed along each chain. So ganon, and 2 new DNA molecules result. Okay, hindi ko anin natin to, but rather punta tayo dito sa may central dogma of the molecular biology or molecularity. The DNA will be converted into RNA through transcription, and then RNA will translate it into protein through the translation natin. Okay, ang portion ng DNA molecule natin which codes for one protein is called a gene or pwede rin ko natin. Mamaya mako natin code doon. And then, yung DNA natin nagre-reside sa may nucleus for protein synthesis and nag-occur siya sa may cytoplasm. So yung DNA nandun sa may nucleus. And then kapag magkaaroon na tayo ng protein synthesis guys through tRNA, mga ganun, or mRNA, mag-occur na siya doon sa may cytoplasm. Okay? So, ganon. So, the genetic code is based on the 3 base sequence in mRNA. And then, each 3 base corresponds to a particular amino acid. So, is 3 base sequence isang amino acids. So, ano ba yung mga amino acids natin dito? Guys, ito yung mga amino acids natin. Okay? Ito yung mga gap. Ito, glycine, alanine, proline, valine, leucine, isoleucine, and so on and so forth, which is the building blocks of protein natin. Okay? So, balik na tayo dito. Ayan. And these are here. Kapag meron na tayong protein synthesis, ito yung mga specific sequence base or codon natin. Kunwari, meron tayong GCA. Ang amino acid na masisynthesize nun is alanine. AGA, and then so on and so forth, arginine. And then AAC and AAU, asparagine. And so forth. So, every 3 bases, sequence base, to 5' to 3', is meron tayong specific amino acids na tinatranslate ng mRNA natin. Kaya nga, translation is a peptide synthesis by ribosome using code from mRNA. So let's consider this sample problem natin dito. sample problem natin. So dito, okay, what sequence of one strand of DNA is complementary to the sequence TAT-GC-AT? Okay, so ito ay naka 5' to TAT-GC-AT to 3'. So kapag complementary guys, yun ay 3'. So ano ba yung natin, di ba? Ang complementary niyan, di ba, sa may by Watson and Crick is A, that is T, that is A. Si A ay para ay T, and si T ay para ay A. Kapag si G naman, that is C, and then G, and then T, and A, that is to 5'. So, tignan natin kung tumama tayo, guys. Although may answer key naman, pero syempre, iba pa rin yung change. O, di ba? Tumama. ATACGTA. Nasaan na? ATACGTA. So, ganon. Next naman natin for the translation. So gagawin naman natin ito guys na proteins natin. Magsisynthesize dito tayo ng amino acids. Kung mapapansin niyo to the process of translation, okay, we have this, okay, for each one natin, 3 bases of nucleotides make one codon. It is a codon responsible for protein synthesis. Di ba? Ito yung mga yon. Ito yung sinasabi ko kanina for each 3. Ayan. And then each codon specifies one amino acid or the genetic code. Ang genetic code natin, guys, is makikita natin mamaya. And then, guys, yung start codon natin, it always start at the August or AUG. And then, nag-i-stop siya sa may uwag. Kahit gaano kahaba pa yan, guys, basta na-detect ng UAG or ano yon, yung mga stop codon natin, di uwaa, uwag, uga. So, dito mag-originate itong means na to. So, ayan, nagkaaroon tayo ng simultaneous polypeptide chain. Yeah! Kaso, ayun, may uwaa, uwag, uga chain tayo, which are the stop codon. So, I'm about to end this whole man's career. So, ayun. No matter kahit gaano kahaba yan, guys, magi-stop at magi-stop yung polypeptide synthesis natin kapag meron tayong stop codon. So, the protein synthesis will stop at the uwaa, uwag, uga. Kaya nga ito yung genetic code natin. Okay? Ito yung pattern natin. So, ito, pinakita ko lang kanina, for every 3 bases or codon, there's a specific amino acid that will be synthesized. Pero kung isa-summarize natin siya, ito yung graph natin. Okay? Magkaaroon tayo ng sample problem natin dito mga guys. Okay, and these are the transcription and translation processes natin. From DNA, okay, di ba? Ito yung strand natin. And then kapag yung complementary strand niya is 3' to 5', nagkaroon ng transcription, okay? mRNA na. Okay, another there, complementary G, TA, CG, CG. And then kapag A na guys, 'di ba? That is U sa may mRNA natin. Hindi na siya T, 'di ba? Uracil, AGPO, and then C, G, and A, and U. And then complementary strand, which is the tRNAs, ayan, U, C, U, CCA, and then CGA. And then kung magkakaroon ng translation processes, guys, dito tayo titingin sa may mRNA. So ano yung aga natin, guys? Arginine daw. Tingnan nga natin. A, first base codon. Tapos second base codon is G. Okay? Ano yung third base codon natin? Is A. So A, G, A. So arginine. So ayan. And then sa glycine natin daw is GGU. Tignan natin. Okay? G, first base. Second base, G. Okay? So nandito. And then U. So glycine natin. So ano yung mga gly na yan? Okay? Balik tayo sa may proteins natin. Kasi nga proteins natin guys is meron silang mga shortcut na pangalan. Ito. Yung mga abbreviation natin, symbol, and meron din silang mga 3-letter symbol. Okay, makita natin yun mamaya. Ayan. Okay. Next naman natin is alanine daw is GCU. So, G, C, and then U. Alanine. So, mga ganun-ganun. Okay? So, kung mapapansin niyo guys, ito nasan siya? Ito, ayan. Ayan, okay. So, remember guys that the DNA is A and U. So, ganun. So, ito means lang to. Kasi nga, di ba? kapag RNA. So, kapag nasa mRNA. So, kung babanat kayo guys, di ba? Tapos yung diniscard is my knowledge about the molecular biology. So, I wish I was admin. So, I can pair with you. Yun lang. Okay. Okay. Punta tayo sa ipa. Sample problem number 3. Ilang ba yung sample problem natin dito? Oh, may answer. Oops. Okay, for our sample problem number 3 natin. Okay, for our sample problem number 3, guys, is, okay, a segment of DNA has the following sequence. Ito. So, 3' na siya, guys. 3' na siya. If 3' na siya, ibig sabihin, okay, ibig sabihin, siya na yung complementary strand na DNA natin. So, sa may A natin, so sa may A natin, guys, what sequence of mRNA bases would appear in the mRNA transcript for this segment? So, kung nasa 3' na siya, guys, ibig sabihin, automatic na ito na yung complementary strand ng DNA natin. Okay, so, A, C, 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 ilang C? 5, 3, 4, 5, tapos 4 na A, and then T, G, T, C, G. Okay. And then sa may mRNA natin, guys, okay, that is to 5' to 3', that is U, di ba, U, G, 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 and then U, 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 A. Sir, bakit A? Kasi nga T, C, A, G, C. That is for 3'. That is for our letter B. Letter C, assume that the first base in this mRNA is the beginning of a codon. What order of amino acids would be translated into a polypeptide? So, kailangan natin dito, guys, yung ating genetic code. Ano ba yung genetic code natin dito? So, itabi natin siya dito. Itabi natin muna. Okay, tabi natin siya. Okay. Na ngayon, ano daw siya, di ba? Per 3 bases, comprised of a codon. So, what is UGG? Hanapin natin UGG. That is tryptophan. Okay? Next, GGG. Okay? G, G, G. That is glycine. Okay? Next is UUU. UUU. That is phenylalanine. And then C, and then, teka lang, parang kulang. Wait lang guys ha. So, wait. Tama ba tayo? 3, 6, 9, 12, 15. Parang kulang. 3, 6, 9, 12, 15. Kulang dito. 3, 1, 3, 6, 9, 12. Wait lang guys ha. May countdown problem. Ulitin ko na lang muna. Okay guys, na-detect ko na yung problem natin. Kulang pala tayo ng isang A dito. Kulang tayo isang A, yun pala ay 4 na A. So, lagyan natin. Then dito is isang U. Sorry siningit ko na lang. So, burahin natin muna yung ating pan natin dito. Okay, and punta na tayo. Okay. So, ito ay UAC. Ano ba UAC natin? So, UAC. That is tyrosine. And then, for the last, that is AGC. AGC. That is arginine. So, therefore, the answer for our letter C is, ang sequence ng amino acids is tryptophan, glycine, phenylalanine, tyrosine, and arginine. Okay, for letter B. Letter A pala yun, sorry. Okay, this one's for letter A, and this one's for letter B. And punta na tayo sa may letter C. Ano daw yung anticodons for each tRNA? So dito tayo babase, guys. For the tRNA natin, for letter C, okay, di ba? Saan ba natin makikita yon? Dito, oh. Ayan. tRNA is the complementary strand of mRNA. So, that is a 3' din. Ganun lang din. So, A, and then 5 C, 1, 2, 3, 4, 5. And then 4 na A, 1, 2, 3, 4. And then isang U, then G, U, C, G. And then 5 prime. So, ayan yung sagot for our letter C natin. Okay, take screenshot guys. So, proceed na tayo. And then next na tayo. Okay. Okay, take screenshot muna. Take a screenshot for this problem. Okay, let's proceed na. So, ayan. Ito. T. T-glype. Ay! Ayun. May nakalimutan na o. UGG. Uwaa, uwag, uga. Uwaa, uwag, uga. Ba't nag-stop ko doon? Uwaa, uwag, uga. Uwaa. Ano? Okay, sorry guys. So, this is just a typographical error. Kasi nga dapat uwaa, uwag, uga siya. Okay, sorry for that po natin. For that time here. So, that's tyrosine and then arginine. Okay. And then for letter C, ay naman yung answers. So for the tRNA, ACCCC, and then apat na A, and then UC, UC, that is supposed to be UG, di ba that UG yun? UUUAC. So A, U is G, G, August, and then UCG, tRNA. Okay. Ito naman yung sa ating sample problem number 5, which will be, syempre, as a part of your learning activity for our ko natin. So mga ganyan natin. And wag niyong kaanin, wag niyong sasagutan itong sample problem number 5 hanggat hindi ko siya pinopost officially as your learning activity. Pero kung nakalimutan kong i-post, okay, swerte niyo. So mga ganon. Okay. Okay, next natin. Okay, another one here is for natin. And then these are the characterization of nucleic acids natin, which we will discuss in our laboratory. Yan yung explanation or the principles behind of your experiment or laboratory activity classes natin. So, mga ganyan. So, thank you for listening, guys, and I hope you learned something. And if you have further questions, kindly PM me na lang sa may messenger kung mayroong confusions na naganap sa inyong klase. And thank you for listening, guys, and yeah, goodbye.